First, I want to thank the meeting organizers for putting together this nice session, and especially having me come to Philadelphia to address you all. So today I'm going to begin by introducing tuft cells as a topic, and then move on to a little bit of a story of my history and how I got into tuft cells, because I started out by studying catfish and ended up studying mouse noses. So it's a kind of a twisted tale. And first, I want to thank and list all the many collaborators who, over the years, participated. Can you see the pointer? No, there is no pointer. Okay. Anyway, and especially I want to thank, on this occasion, Bob Margolskee, who provided research materials to us. Okay, Bob Margolskee, who provided. And Bob has over the years provided research materials to a huge number of investigators, and not only is his intellectual discoveries important for the history of taste research, but his generosity in sharing research materials has been a real boon to the field. Okay, so we and other complex organisms have an intrinsic problem of we need to keep the outside out and keep our insides in, and you can't have passive diffusion of things across a barrier. So we have epithelia, and the epithelial barrier has tight junctions across the top that are a nice barrier to most ions and to any soluble substances. And the question then arises of how, as an organism, can you detect chemicals and respond to chemicals in the outside world if you have this epithelial barrier? There needs to be some way of sensing chemicals in the world. And there's several ways that we and other organisms have solved this problem. The first is that not all chemicals are prevented from penetrating the epithelium. Lipid-soluble substances, for instance. So hot pepper extract, capsaicin, is lipid-soluble. It just goes right through the epithelial layers and can penetrate into the tissue below. And so all you need to do to detect any lipid-soluble substances is hook a nerve fiber up, comes to the surface of the epithelium, have a receptor on the nerve fiber to respond to that chemical, and you have a response. So for lipid-soluble substances, there's not really an intrinsic barrier. But what about the water-soluble substances? And for those, nature has invented two different sensory systems that you're very familiar with. The olfactory system, which is a collection of neurons that have an apical dendritic process extending to the outside world so it can contact these water-soluble substances, and taste buds, which are not neurons. They're specialized epithelial cells. The top of a taste bud extends out through the taste pore to contact the contents in the saliva, and then the taste buds form a peripheral synapse onto the first nerve process, carrying the information to the brain. And the taste receptors cells, many of them use a canonical taste receptor cascade that Bob Margolskee was instrumental in defining for us, and it includes a family of GPCR taste receptors, gusteucin and other G proteins, and finally a channel TRPM5 that generates helps generate the action potential in the cell. So this cascade, this taste receptor cascade, is an important hallmark of taste receptor cells, and as it turns out, also of tuft cells elsewhere in the body. So, and that brings us to the question of tuft cells. So we and others have described a population of cells that are now called tuft cells that are specialized epithelial cells. Their apex sticks up through the epithelial tight junction layer, so they are in contact with the external fluids. And they use the taste transduction cascade to detect and respond to these substances. Some of them are hooked up to nerve fibers, others aren't. The term tuft cells has now been broadened. Originally, tuft cells were first described on the basis of light microscopy in the 1920s. Light microscopy of gut showed this very strange cell type that had a tuft of apical microvilli, and nobody knew functionally very much for these. And then with the advent of electron microscopy, these cells were then better characterized, And then people started finding other cells that look kind of like tuft cells, but they didn't have a bush of microvilli. What they had was a brush, very stiff kind of microvilli with a lot of actin in them. They were called brush cells. And then we also found other cell types throughout. And this class of cells, which now have been given the name tuft cells, even though technically they aren't tuft cells, are found throughout the whole body. So you find some in the airways, you find some in the gut, you find some in endocrine organs, et cetera. And these can be easily identified now thanks to Bob Margolskee and his green mice, because he has developed two lines of mice, one Gustusin GFP, another TRPM5 GFP, that help identify these cells. And the pictures on this screen show you some of those cells throughout the body. So what do we got there? At the advent of electron microscopy, and shortly thereafter, investigator Mary Whittier, who's shown in the upper right here, in England, started describing a class of cells in teleost fishes that were, she called solitary chemosensory cells, that have some of the characteristics of tuft cells morphologically. They're innervated. Mary Whittier has not been recognized suitably in the scientific annals because she was a woman working in England in the 1960s. Need I say more? She was married to a famous biologist, and so she really didn't need the recognition herself. So what she described then are these cells. This is a picture from her paper in 1970. These are isolated cells in the epidermis of fish. You can see they have a single process extending outside the epithelial boundary. Here, for comparison, is a taste bud. She described that these cells are innervated. She found them in many, many different species of fish. In fact, all non-mammalian aquatic vertebrates show these kinds of cells in their epithelia. And looked at here in EM, some of them have multiple villi sticking out. This is a scanning electron micrograph of the surface of the epithelium showing these cells in zebrafish. This is more recent work. But they had a very widespread distribution phylogenetically and also widespread across the fish. So they were covering the surface of the fish. There are far more of what she terms solitary chemosensory cells than there are taste buds on the surface of a fish, even catfish. So here's a typical picture of what they might look like, a surface view that she drew of this, where you can see a top of a taste bud here, the taste pore, and then just scattered in the epithelium are all these many solitary chemosensory cells. This was, again, 1971. So many years later, I was interested in fish as a graduate student. The first half of my career was studying fish neurobiology and sensory systems in fish. And of course, these were a fascinating subject for research since we didn't really know what they did or what they were. But a group at Monell here was studying catfish taste buds. And this is before the cloning of the catfish taste receptor. Catfish taste receptors. And this is including Bruce Bryant, John Teeter, Bob Kagan, Joe Gran, Lynn Kalinowski. Here's the three pictures I could fish off the web really fast, showing them. And what they were doing is they were trying to biochemically isolate taste receptors in catfish. We all knew catfish had very high affinity taste receptor systems, much higher affinity than any mammalian taste receptor. And so they thought they could use biochemical means to isolate the fish. They identified two probes, one for an arginine receptor, one for an alanine receptor. And I said, oh, you know, guys, it's kind of interesting. If you have a high affinity probe for an arginine receptor, we could just apply it to the surface of the epithelium and see where it's localized. And they said, great. And so we had this collaboration. And here's a picture from our paper. You can compare it to this one. Here's the top of taste buds, just like taste buds here. And then in between the taste buds are all these little dots. And those are the solitary chemosensory cells. So this is the first demonstration that a solitary chemosensory cell, I guess you'd say tuft cell, shares some components of taste transduction with taste buds. And then we got to wondering, well, if you look across the entire range of fishes, from hagfish to lampreys to teleosts, They all have solitary chemosensory cells scattered in the epithelium, in the nasal cavity, in the respiratory passages. Did these all just disappear when mammals formed, when we came onto a land transition? It seems unlikely. There are still passageways in our body that are kind of wet. Maybe these cells have persisted into that. And so we began a collaboration with several investigators, including Wayne Silver, who was at Monell in those days, on looking for these kind of cell in the nasal epithelium of mammals. And for that purpose, we use some of the tools then available to us from Bob Margolskee, including antibodies to gusducin. And this is a picture of a cell in the nasal epithelium of a rat with showing gusducin immune reactivity. And it kind of looks, you go, oh, wow, that kind of looks like a solitary chemosensory cell. And I wonder if there's more of them. How hard are they to find? And it turns out they're all over the place. They're scattered throughout the entire respiratory epithelium in the nasal cavity. There's none in the olfactory epithelium. There's other cells that share some of the signaling properties, but none of these. And we were using gusducin immunoreactivity to locate these cells at the time. And then Bob invented a mouse, a gusducin GFP mouse, so the cells now were glowing green in the dark, and so then we could use his mouse to identify them. And you can see there's a lot of these. This is not a rare cell type in the epithelium. So then we go, well, that's kind of interesting. Mary Whittier said these things extend to the surface of the epithelium and are innervated, so we looked for that. So here's a picture from EM showing that they have apical microvilli sticking up to the surface of the epithelium. And then, whoops, we wondered what the cells are doing. So we used some probes that I got from Charles Zucker's lab to show that these cells express the bitter receptor messenger RNA. So they're expressing the T2R class of receptor. And they're innervated by nerve fibers. We could show using antibody probes to various nerve fiber components. And not only are they innervated by nerve fibers, they're innervated by a special class of nerve fibers that expresses CGRP as a peptide marker and substance P. They're polymodal nociceptors. So these are pain fibers in the nasal epithelium innervating these solitary chemosensory cells. We can show that they make synaptic contact. There's a synapse of a solitary cell onto a nerve fiber. And what does it mean then functionally? If you can activate the nerve fibers in the nasal cavity, first of all, what do they do? And secondly, can you activate them through these cells? So again, this is a collaboration with Wayne Silver. So first, we tested, what do you do if you activate the nerve fibers directly? And so the assay we used is a respiratory response, was pioneered by Wayne Silver with Russ Mason and Joel Marignac. And this is a record of breathing in a mouse. So each up and down mark is one breath. And this is a indication of the time between breaths. So before stimulus is applied, you have a fairly constant respiratory rate. You put on the stimulus, and basically the animal holds its breath for a little while. So you wash off the stimulus, and then it comes back and returns to near baseline level. So that's what these records show. That if we put on capsaicin, which you remember directly activates the nerve fibers, then you can cause a respiratory pause by the animal. So then we could ask, well, what happens when we put bitter stimulus on these cells and on their nasal respiratory tract? And so this is a graph of single cell responses done in collaboration with Sue Kinnaman, who will be giving closing remarks after dinner tonight. And this is imaging from a single cell labeled with GFP, thanks to Bob, and showing responses, calcium responses in the cell following stimulation with denatonium and PTC, two well-known bitter compounds. And then if you do and just flow denatonium across the surface of the nasal epithelium, you get a respiratory record that looks like this, pretty much identical to when you directly activate the nerve fibers with the capsaicin. So yes, the SCCs express bitter receptors. They're functional. They activate the cell, and then the cell can activate the nerve. So then we got to asking, well, why would you build a system that responds to bitter substances in the nose? It's not often that when you're picking your nose, you first dip your finger in denatonium before you put it in your nose. So what are these? What's the natural ligands for these things? And we were kind of puzzled for a while. And then Marco Tizzano joined the lab. He's often referred to as the madman from Verona. But he came with the idea, oh, well, of course, these cells are activated by bacterial byproducts, by the quorum sensing molecules of gram-negative bacteria. And we go, what? That's impossible. That can't possibly be right. And he said, yeah. Try it. You'll see. And so we set him the task of trying it. And sure enough, these cells respond to these two products, ESI and LASI, that are the molecules produced by the gram-negative bacteria to signal to each other their colony density. So the bacteria form a biofilm when they get to be a high enough density. And these are the two signal molecules used by gram-negative bacteria to do that. And sure enough, we see the cellular responses to these gram-negative bacteria signals. You can figure out a dose response curve. The range that they're responding to is about the range where you expect the bacterial signaling to occur in the nasal epithelium. So that was kind of interesting. And then when we flowed these compounds across the epithelium, sure enough, they cause respiratory depression. So the cells in the nasal cavity are using T2R receptors to respond to natural ligands produced by bacteria. So here's the model we have, that in the nasal epithelium, we have two modes of activating the nerve fibers. One is a direct mode, for instance, capsaicin or acetic acid, both of which can penetrate the tight junctional barrier, can directly activate the nerve fibers. And the alternate path is to what we were calling solitary chemosensory cells in those days. And any T2R agonists, like denatonium or the bacterial products, activate these cells. It requires the intact taste signaling cascade using T2Rs, gasteucin, PLC beta, and TRPM5 that we were able to demonstrate using knockout models provided to us by Bob Margolskee. And then these nerve fibers are polymodal nociceptors, the pain nerve fibers that convey the message to the brain. But we also realized, polymodal nociceptors are interesting because they don't just convey the message to the brain. That a better model would be looking like this, where they carry the message to the brain, but then they also have local terminals all through the epithelium and mucosa, wrapping around blood vessels, potentially activating mast cells. And we know that these two peptides, CGRP and substance P, are necessary for the action on the blood vessels, where they cause opening of the endothelial cells for plasma leakage. So they allow plasma out into the surrounding tissue, and also degranulation of the mast cells. So these are the hallmarks of an innate immune response in the tissue. So we could test that. We said, well, do they cause plasma leakage? Well, how do you measure plasma leakage? It's pretty straightforward. You take a fluorescent substance coupled to albumin or something big, inject it intravenously, it runs through the animal. Under normal conditions, none of that should get out into the surrounding tissue because the vessels are tight. But if you open the tight junctions in the vessels, it'll leak out into the plasma, into the surrounding tissue, and you'll see fluorescence. So here's our experiment. So here's from a wild type mouse, and we inject the fluorescent dye on one side of the nose, we flow in denatonium or another bitter activator. On the other side, you leave it intact, and then you just split the head in half and visualize it. And on one side, you see fluorescence, meaning there's been plasma leakage. On the other side, none. And you can measure this just, how bright is it? And you get a graph that looks like this. So that's the difference in fluorescence with the denatonium. If you use a gasteucin knockout mouse interfering with the taste transduction pathway, you don't see that effect. So it's dependent on the T2R signaling. TRPM5 discount, same thing. Whereas if you use capsaicin, which remember directly activates the nerve fibers, those drugs have no effect. So you can still get the effect of plasma leakage even using the knockout mice, but only if you directly activate the nerve fibers. So again, this model seems to hold. Let's see what else. And we then determined, based on a suggestion from Jake Sanders, then a graduate student in the lab, that this is cholinergic signaling. That is, the SCCs, tuft cells, are releasing acetylcholine. The nerve fibers are using nicotinic receptors to generate their response. So when we use blockers of the cholinergic signaling, you see you can decrease the plasma leakage. And we can similarly show the same thing with mast cells. So here's a mast cell stained with a blue dye. Under normal conditions, they're nice and condensed. And then when you activate them, they spray out this halo of little dark granules that are their substance inside. So you can just measure how many mast cells are showing this degranulation and how many are intact. And you can show signaling is necessary in wild-type mice. Then you put the denatonium on and you get an increase in degranulation. Here's the baseline level. Knockout mice don't show this. Knockout tryptophan 5 knockouts. And the capsaicin induction, or sorry, the mast, and you can put on a drug that will directly degranulate mast cells. They all degranulate. And then you can show using cholinergic blockers that this effect on the mast cells is necessary for cholinergic transmission to occur. So the transmission of the message from the SCC to the nerve fiber, which is cholinergic dependent, is required for this effect on mast cells. And it's also dependent on substance P, one of the peptides I told you about in the signaling pathway. So in summary, what we've established then over the many years of work was that the solitary chemosensory cells, now called tuft cells, in the nasal cavity are using a taste transduction cascade of T2Rs, glycogen, PLC, tryptophan 5 to generate a response to exogenously applied chemicals. These cells then release acetylcholine, activate the polymodal nociceptive nerve fibers, which in turn release peptides into the surrounding tissue. The peptide release then causes a innate immune response involving plasma leakage and mast cell degranulation, and probably many other things that you will hear about today later on. So I just want to wrap up giving you an overview that we discovered these cells because I was working on fish, and we just got to the question of, well, is this a system unique to fish, or could we see remnants of this system in terrestrial vertebrates? And the answer is, we still don't know for sure the answer to that question, whether there is phylogenetic continuity between what we see in fish and what we see in mammals. But nonetheless, it led us to the discovery of these so-called tuft cells, solitary chemosensory cells in the nasal epithelium. So with thanks to both Bob Margolskee and Mary Whittier, thank you for your attention.
Questions? I just have a simple question. Are among the ligands for the tuft cells, are many of them or any of them volatile, or most of them just are not volatile?
Well,
how they get to the respiratory epithelium.
Well, so in air-breathing animals, things that are volatile still have to diffuse and enter into the mucus layer, so nothing is in air when they're detected in the nasal cavity anyway. So I think that there are many volatile substances that will partition into the mucus layer and then activate these cells, but I don't have direct evidence of that. I think some of the later speakers might address that issue.
Thank you very much. You know, I admire your work, and it inspired also our work about your concept that they are connected to polymodal nociceptors. You and I and all the others in the field draw it like this. But is there really evidence that it is one and the same nerve fiber that responds to capsaicin and has a branch toward SCC? Or are these two separate types of nerve fibers? For example, you have a good antibody for TRPV1 to show that these fibers at the tuft cells or SCC are also TRPV1 positive, or are these separate populations?
I'm trying to remember. So we didn't do that experiment, except we know that fibers that display CGRP, substance P immune reactivity are considered to be polymodal nociceptors throughout the body, right? I think someone, it may have been even Zucker's lab, did a ablation of that population with racinifer toxin, but they didn't test, I don't think they tested the SCC component of that. So I don't think I have a definitive answer. We have circumstantial evidence that because of the peptide presence that they certainly are included in that class of fiber. We see branches from the fibers that are invading the SCCs running out into the submucosal layer. So it's the best I can do.
So Tom, I want to go back to the fish, and I'm going to show my ignorance in fish physiology, but you demonstrated that these tuft cells in the respiratory system create an apneic response. Basically, if you inhale a nose full of bacteria, you don't want to aspirate it into your lungs. The fish, and I know nothing about gills, when you activate these chemosensory cells on the fish skin, the fish can breathe. Is there an apneic response to the fish, or what are they actually stimulating there? Because they're not in the airway; they're actually on the surface. So, what's the role of these sensory cells on the skin fish?
So, do fish breathe? Is one of the questions I got from our animal welfare committee. One of my and fish do indeed breathe. You can see their operculi moving. But the solitary chemosensory cells in fish are not restricted to the airways. They're all over. Any external surface touching water has these cells. So they're on the tail, they're on things. So I think in fish, they're not tightly linked to respiration, which brings up a related question of, so what are they doing in fish, right? And we don't even know that they express T2Rs in fish. We don't know much about solitary chemosensory cells in fish. Keiko Abe's lab has described T1R family receptors in fish and said they're not in solitary chemosensory cells. We have some behavioral evidence and physiological evidence from a specialized marine fish called sea robins that have high density of these solitary cells on their fin rays. And they're used in the context of feeding, so they're used to detect food. The only other report of behavior related to these cells is in a codfish relative called a rockling, where the investigators, Kurt Kadeshal and Steve, can't get the other name, but said, oh, they're used for predator avoidance. So we have no evidence of what they're really doing in fish, and it's one of the things I'm trying to follow up on now in zebrafish, looking at what are the cell populations doing there. So it's a great question. They definitely don't cause respiratory depression, though.
One word.
Is there any evidence for that these cells may play a role in mechanosensation?
I don't know of any evidence that that is the case, but there's no evidence that they don't play a role in mechanosensation. Most electron microscopists think that cells with floppy microvilli are not mechanoreceptive particularly. I mean, any cell is going to be mechanoreceptive if you hit it hard enough, but the mechanoreceptive single cells in the epithelium usually have a very stiff brush of microvilli, like the brush cells in the trachea are thought to be mechanosensitive.